Microbiology, Chapter 1, Section 1. Section 1, Microorganisms, Tiny Titans of the Earth, to be specific. When we talk microorganisms or microbes, okay, we're talking about things that, for all intents and purposes, are going to be too small to see with the naked eye. Now, in our hubris, we think because we are large multicellular things that these tiny microbes, these single-cell organisms, whether they be prokaryotic bacteria, archaea, or as we're going to see in this course, protozoa, single-cell yeast, fungi, things like that, because they're so much smaller, that must mean that they can't do much. There's just not much happening there. When in actuality, well, no, they're actually quite diverse in form and function. As we're going to see, even the bacteria, the single-cell organisms, have a lot going on metabolically. They are very diverse in not only their structures, but their abilities. We're going to see that many of these microorganisms, specifically archaea and prokaryotic bacteria, pole to pole, North Pole to South Pole, every place in between, you're going to find these guys. I like to joke that the archaea, you're going to find every place we're not. Sulfur hot springs down at the bottom of the ocean, in the sulfur vents. Supposedly that they even isolated archaea growing in nuclear reactors. I'd like to see that. That would be kind of cool. These single-cell organisms, well, as we're going to see, it's every cell for itself. It's not like us. Yeah, you're sitting there as you're watching this. You're trillions and trillions of cells. But they're working together. They take on distinct roles within a tissue, the tissues within an organ systems, organism. No, these single-cell organisms, it's every cell for itself, but they may come together towards a common goal. But still, every cell for itself. They're going to form microbial communities. This is now where research is really starting to look. When I was an undergraduate in the 90s, we're looking at, individual cells. That's what we were trying to do. Let's study individual cells. Let's study individual species. What's going on here? And we're finding out that nowhere on this planet is there an individual species, a pure culture, except in the laboratory setting. So if you really want to know how changes in the environment, changes in, the signals that a cell gets, whether it's a bacteria, fungal, protozoan, well, you have to have it in a mix, a community. Not only a community composed of the same species, but a mix of species that are there because, even though the cells within the same species are in competition, they're also more in competition with outside species. So when you go and you look, at slides of environmental samples, or you look at slides, as you see here, granted, keep in mind this is an electron microscope picture that was computer colorized. So no, your microbes in your gut aren't this colors. Nowhere, whether it's a pond sample, I think that's what a pond sample, or your microflora of your gut biome, nowhere is it an individual cell type. It's a mix of different cells, and even the ones here that look alike, well, they may just look alike, but they're still different species forming a community in competition and at the same time still working together. These old microorganisms, these single cell things, are some of the oldest forms of life on this planet. Prokaryotes, prokaryote, before the nucleus, before organelles. They are the oldest. They've been here a couple billion years longer than we eukaryotes. They're also the major fraction of the Earth's biomass. Okay, there are more of them than there are of us. They surround plants and animals. You're covered in bacteria inside and out. So is your dog, your cat at home, your goldfish, the plants and things like that. We have evolved in their presence, to be covered by them, to have them around us. They affect us humans. Diseases. They're in our food, our water, they're in our soil. We want them there. Yeah, in our food and our water, we may not want them. We want clean water, we want clean food. But, hey, I don't know about you guys, but I like some sauerkraut on my hot dogs. A beer once in a while. I'm sure you know what I'm talking about. Those of you who are over 21, thank you. Bread, stuff like that. Infectious diseases. Turns out, as we're going to see later chapters, we are some of the cleanest humans in history. We're also still some of the sickest. Maybe we're too clean. Maybe that hygiene hypothesis. We'll see. So when you look at it, there's this interplay of microbes all around us. Human health, our ecosystem. Think about it. Tree falls in the forest. What happens to the tree? Well, the tree breaks down, the tree decays, the tree becomes new carbon and nitrogen sources for other plants. Well, how does it decay? Microbes, fungi, bacteria, break it down. Our health, animal health. Turns out agriculture, as we're going to see and talk about in later chapters. For those of you who have never been on a farm, turns out farmers, I believe at least once a year, send a soil sample off to a survey office. Tell us what the phosphate, the nitrogen, all the mineral concentrations are. That's how they determine which fertilizers to spray on the fields. Well, guess what? They're starting to try to get away from that. Instead of, spraying, more and more nitrogen, which can then leach off the field and get into the surrounding waters, why not start spraying bacteria, microbes? Add more nitrogen fixing microbes into the soil, more nitrogen in the soil. Win-win. Now, in this chapter, we're going to be talking about some different tools you need. Different types of microscopes. Cultures, okay? Yes. Even though in the real world, all microbes are going to be growing in mixed cultures and things like that, but in the lab, we want them pure culture, so we can sit there and know exactly what that microbe, what type of nutrient media it needs to grow in or on. How do they grow? What's the ways they do this? How we can visualize them. You're going to hear me using terms, and it turns out that there's a lot of terms that are in microbiology you're not going to see in a lot of different places. One of them is colony. Okay? This is what's called a streak plate. Here, what looks like just one big thing. Well, it turns out you have these individual spots, these colonies. Here, an individual colony you see as a dot, but when you magnify that dot, it's millions and millions of individual bacteria. That's a colony. What does it have to do with this blanket right here? Well, this streak plate, you have so many colonies right here in this area that they are all growing, touching each other. Well, can't do anything with that. So you streak around here, and then you take some of that and you streak it here. And you look here, you can see that where this is all one flat growth, because all hundreds and hundreds of colonies all growing on top of each other, you can start to see where there's some separation. There's, one or two logs less here. Well, you take some of this and you smear it here. Now you get these individual spots, these individual colonies. And this I can do research with in the lab. This is what you're going to be doing. This is what you can do research in the lab. Can't do it with this. Can't do it with this because the colonies are all touching. How can I verify that I'm getting one type of microbe, one type of bacteria? I can't, because they're all growing on top of each other. There could be a contaminant in there, but it's overgrown. It has stuff growing on top of it so that I can't see. And it turns out, yeah, when you get into a lab and we start doing these and you compare the growth characteristics of different bacteria, Staphylococcus aureus, Escherichia coli, yeah, the colonies don't day glow bluish green, teal like these do, but they'll have different colors, different growth characteristics. You can't sit there and look at it and say, oh, this is E. coli, this is staph. No. But you can look at two of them on the same plate and go, yeah, these are two different things. I need to be careful which one I pick and which ones I don't. And then at the end of the chapter, we're going to be talking about molecular biology and the genomics.